Our today's topic is structure of chromosomes. In our previous lecture, we have studied that chromosomes are carriers of genetic material and there are 46 chromosomes, and these 46 chromosomes, they are arranged in 23 pairs. Out of 23 pairs, 22 pair of chromosomes are called as autosomes, and only one pair of chromosome, it is called as allosome or sex chromosome. The autosomes are mainly concerned with determination of body characters, whereas the sex chromosomes, they are responsible for determination of sex. Now we'll see the arrangement of chromosomes. This is the ideogram showing arrangement of chromosomes according to their decreasing size. See the size of chromosome. This is first pair of chromosome. This is 22nd pair of chromosome. The first pair of chromosome is very long, whereas the 22nd pair of autosome, it is very short. But keeping in mind the position of centromere, if we look carefully, the position of centromere is aligned or it is in a straight line. This is the last pair of chromosome, that is 23rd pair, and it is called as sex chromosome, out of which one is called as X chromosome and the other one is called as Y chromosome. But this is the diagram of a single chromosome. The single chromosome, it appears like a ribbon. This is before replication and this diagram is after replication. In this diagram, we'll see different parts such as telomere, chromomeres, secondary constriction, centromere, secondary constriction or nucleolar organizer, and satellite. Now each chromosome, it has two chromatids. So this is a replicated chromosome. This is one half and this is the other half of the chromosome. This each half of chromosome, it is called as chromatid. The outer covering or the membrane of chromosome, it is called as pellicle, which consists of matrix. This is the central part of the chromosome, and this central part, it is called as centromere. In this diagram, centromere is present here. This is the centromere. The centromere is also called as primary constriction. The centromere, it helps to join the chromatids. These are the chromatids and this is the centromere. So with the help of this centromere, the chromatids are joined with each other. Centromere or primary constriction, this is nothing but a narrow region which is present in the chromosome. Besides primary constriction, the chromosome may contain one or more constrictions called as secondary constriction. So these are secondary constrictions. Now this part, this is called as secondary constriction 1, or it is also called as nucleolar organizer. The name itself suggests this is a place or site where nucleolus is organized. Talking about secondary constriction, again this is also a narrow region present in the chromosome, but it is mainly concerned with synthesis of rRNA. This chromatid, it consists of highly coiled double-stranded DNA. So this is the DNA that is present in the chromatid and it is called as chromonema or chromonemata. Or the central coiled part of chromosome, it is called as chromonema or chromonemata. Then comes the next part of chromosome which is called as chromomeres. Chromomeres are nothing but a group of bead-like granules. All these are group of bead-like granules which are present on chromonemata are called as chromomeres, or chromomeres are the regions of highly superimposed coils, whereas chromonema, this can also be called as gene bearing portion of chromosome. Telomere the end part of chromosome, it is called as telomere. And if the chromosome is having satellite, then that type of chromosome is called as satellite chromosomes. So these are the different parts of the chromosomes. In all, there are 8 parts of chromosomes, which are as follows. The first part, it is called as chromatids. Each chromosome consists of 2 chromatids. Centromere. Centromere is a part of the chromosome which helps to join the chromatids, and it consists of a disc-like plate called as kinetochore. And the most important role of this kinetochore is spindle fibers are attached at kinetochore during the process of cell division. Secondary constriction or nucleolar organizer, it is the place where nucleolus is organized. Chromonema or chromonemata, each chromatid consists of double-stranded DNA called as chromonema. Chromomeres, a group of bead-like granules present on chromonemata, are called as chromomeres. Satellite or satellite body, when satellite is present along with chromosome, then that chromosome is called as satellite chromosome. Secondary constriction 2, it is mainly concerned with synthesis of rRNA, and telomere, it is the end part of the chromosome. Now, depending on the position of centromere, there are 4 different types of chromosomes called as metacentric, submetacentric, acrocentric, and telocentric. Now, in all these chromosomes, we just have to remember the position of centromere where it is located. In the first type of chromosome, that is metacentric chromosome, the centromere is present exactly in the center of the chromosome. So this is centromere, and these are the arms of the chromosomes. So since it is present in the center, the arms of the chromosomes are of equal length. So when the centromere is present in the center, then that type of chromosome is called as metacentric chromosome. This is the part where the chromosome can bend. That is, centromere is the only part in the chromosome where the chromosome can bend during the process of anaphase, due to which it appears as English alphabet V shape. So this type of chromosome, it appears like English alphabet V shape. Then comes second one, that is submetacentric. In this type of chromosome, the centromere is present slightly below the center. So due to which the arms of the chromosomes are unequal, one is long and the other one is short, because the centromere is present below the center, due to which the chromosome, it appears like English alphabet L. Third type is called as acrocentric. This is third one. In this type of chromosome, the centromere is present towards the end of the chromosome, due to which one arm is very long and one arm is very short, and it appears like English alphabet J. The last one, telocentric. In this type of chromosome, the centromere is present at the end of the chromosome. Hence, this is called as telocentric chromosome. The most important point in this type of chromosome is the chromosome has only one arm, because the centromere is present at the end of the chromosome and it appears as English alphabet I. So this is the last type and it is called as telocentric. Only thing is this dot, it is present very close. Hence it appears as I shape. So on the basis of position of centromere, the 4 types of chromosomes are metacentric, in which centromere is present exactly in the center. Submetacentric, the centromere is present just below the center. In acrocentric, the centromere is present towards the end of the chromosome, and in telocentric, the centromere is present at the end of the chromosome. So these are the different types of chromosomes. Now these are sex chromosomes. As we have already said, in sex chromosome, one is X chromosome. So this chromosome, it is X chromosome, and this chromosome, it is Y chromosome. Now in X chromosome and Y chromosome, there are certain point of differences. The first one is, if we carefully look at the chromosomes, see the length of the chromosome, the X chromosome is very long, whereas Y chromosome is very short. So this is longer and this is shorter chromosome. If we see the position of centromere, X chromosome is metacentric as the centromere is present in the center and both the arms are equal in length. Y chromosome, it is acrocentric because the centromere is present towards the end of the chromosome. Then both the chromosomes, it shows homologous region and non-homologous region. Homologous region, they contain similar genes, whereas these are non-homologous regions, they contain dissimilar genes. The non-homologous region contains X-linked genes on X chromosome, and non-homologous region of Y chromosome contains Y-linked genes. X chromosome contains large amount of euchromatin and small amount of heterochromatin, whereas Y chromosome, it is exactly opposite to X chromosome. It contains small amount of euchromatin and large amount of heterochromatin. X euchromatin contains large amount of DNA, hence it is genetically active, whereas euchromatin contains less DNA, hence it is genetically less active. So these are few of the differences. And one more thing, the most important process, that is crossing over, it takes place in homologous region of the chromosomes. So these are the sex chromosome, this is X chromosome and this is Y chromosome. Let us revise the differences that we have taken. X chromosome, it is longer, Y chromosome, it is shorter. See, it is comparatively longer and shorter. Then the next point of difference is X chromosome, it is metacentric and Y chromosome, it is acrocentric. This is on the basis of position of centromere. Euchromatin is large, heterochromatin is small. Whereas in Y chromosome, euchromatin is small and heterochromatin is large. Both are exactly opposite to each other. This is genetically active, this is genetically inactive. As it contains more DNA, it contains less DNA. The non-homologous region is long in X chromosome, hence more number of genes are present in X chromosome. Whereas non-homologous region of Y chromosome is small, hence it contains less genes. X chromosome contains X-linked genes and Y chromosome contains Y-linked genes on the non-homologous regions. So this is all about the chromosomes. Thank you.